Hello everyone, welcome back to Immune Biology. Let's proceed with the next class. In this class, I'll be having a discussion on humoral immunity or antibody mediated immunity. As we discussed in the last lecture, acquired immunity is of two types. The first type is cell mediated immunity or CMI, then that is by the virtue of some T lymphocytes, and the second one was antibody mediated immunity, AMI, or also called as humoral immunity. Why is this called as humoral immunity? Because the word humor means fluid, and because all the antibodies that are responsible for AMI are present in the mostly in the blood, so this is called as humoral immunity. CMI is because of T cells, T lymphocytes or thymocytes, and AMI is because of B lymphocytes or B cells. Now as you can see in this picture, this is a B cell which gets differentiated and forms a plasma cell. Plasma cell is a type of B cell, and these plasma cells are now responsible for formation of antibodies. These all are antibodies, these Y-shaped molecules. We'll be having a healthy discussion over antibodies, plasma cells, immunoglobulins about in this video. Let's start with the B cells. You see, B cells are of two types. The first type is the plasma B cell and the second is the memory B cell. Now plasma B cells are responsible for the formation of antibodies, so they are called as antibody factories, and memory B cells, as the name implies, are responsible for the secondary immune response. It would memorize the first encounter it has done with the antigen, and in the second encounter with the same antigen it would convert immediately into the effector cells, plasma B cells, and would produce antibodies, which is called as secondary immune response. Now this is a word called antibody. What is an antibody? It is a protein substance which is produced against an antigen. Whenever a foreign antigen enters your body, your plasma B cells would produce certain glycoproteins called as antibodies, which would then interact with those antigens. Now there is a term called as immunoglobulins. So what are immunoglobulins? Are immunoglobulin and antibody synonyms of each other, or is there any difference? See, there is a slight difference between immunoglobulin and antibody, and the difference is that the immunoglobulins are the antibodies which are keep on circulating in the body fluids. In short, those antibodies which are present in the body fluids are called as immunoglobulins. Other antibodies are also present on the surface of some lymphocytes, so they are not called as immunoglobulins. Now immunoglobulins in short is called as Ig, and there are 5 types of immunoglobulins which we need to study. The structure of immunoglobulin and antibodies is almost same. These are of 5 types: IgG, IgA, IgM, IgE, and IgD, and a mnemonic to remember all of them is called as GAMED. By the word GAMED you can remember the 5 types of antibodies. Now the question is, why are they called so? Why are they named so? So I'll be discussing about this in a while. Let's first start with the structure of immunoglobulin, structure of Ig. As we discussed, it is a glycoprotein present in blood plasma, and it is made up of 4 polypeptide chains, 2 heavy chains and 2 light chains. So it is called as H2L2 molecules because of the number of polypeptide chains present in them. This is a basic structure. It looks like a Y-shaped molecule. Now in this figure there are 4 polypeptide chains, as you can see, 2 blue chains and 2 green chains. Now the chains on the outer side, the green ones, are smaller, so they are called as light chains, and the one on the inner side are longer, so they are called as heavy chain or long chain, and the ones on the outer side are called as light chain or short chain. These chains are joined together by disulfide bonds. These are disulfide bonds, as you can see in the picture. These all are disulfide bonds which connects the 2 types of chains. Now on a light chain, as you can see, I have demarcated the area. One in the dark green is called as the variable region, and one in the lighter green is called as the constant region. Now this variable region of light chain is responsible for the specificity of antibody. The sequence, the amino acid sequence, because it is a polypeptide, so it would be made up of amino acids, so the amino acid sequence on the variable chain varies with the antibody. Every antibody has got its own polypeptide sequence, own amino acid sequence in this region, so this is called as variable region. It is different for different antibodies, and it is responsible for the specificity of antibodies. The other one is called as the constant region of light chain, and it does not change. Similarly, on the heavy chains also, Similar regions are there: variable region of heavy chain and constant region of heavy chain. Variable region is responsible for the specificity of antibody, as it is different in different antibody, and the other one is the constant region, which is same. Now as you can see in this picture, the entire structure which is present in this bigger circle is the constant region, and the region inside these smaller circle, these two circles, are the variable regions of both light chain and the dark chain. Now the most important question: the site where antigen gets attached to antibody. This is called as antigen binding site. Remember, an antigen would bind where a heavy chain meets with the light chains. At this site antigen would bind. So an antibody would have two antigen binding sites, and these antigen binding sites are called as paratope. So what is a paratope? A paratope is actually the antigen binding site, where the site on the antibody where an antigen attaches to antibody is called as paratope. I'll take two words here, epitope and paratope. Listen to this carefully. See this figure. This is an antigen. This is an antigen. Now this part of antigen, this blue part of antigen would interact with the antibody. So the part of antigen that interacts with the antibody, that is attached to the antibody, is called as epitope. Similarly, the part of the antibody which is attached to the antigen is called as paratope. So basically an epitope attaches to a paratope. A paratope of antibody interacts with the epitope of antigen. The entire antibody and the entire antigen do not interact with each other. Antigen's epitope and antibody's paratope interact with each other. Now we will have a discussion on how antibodies work in the body, what are the functions of antibodies. The first thing that we know is the antibodies interact with the antigens, but there are different mechanisms by which these antibodies would interact with the antigen, and the first mechanism is neutralization. As the word suggests, such antibodies directly neutralizes the antigens that are present, that enters your body. These would neutralize the toxins which are released by the pathogens. Many pathogens, and the best example is the pathogen Clostridium tetani. When Clostridium tetani, that causes tetanus, when this bacteria enters your body, it releases two toxins called as tetanospasmin and tetanolysin, and these toxins are responsible for the effects that are produced in tetanus. So your antibodies against the antigens of Clostridium tetani, the toxins of tetanospasmin and tetanospasmin and tetanolysin, so these antibodies would neutralize these toxins. So such antibodies are called as antitoxins. That means the toxins, the antibodies which performs the action of neutralization are called as antitoxins. Now in case of snake bite, when such patients come to the hospitals, what doctors do, they administer these patients with antitoxins, because in a snake bite case, a snake would always release its toxins into the body, and we administer the patient with the antitoxins, which are kind of antibodies. So this is the first mechanism by which an antibody would interact with the antigen. The second type is agglutination. Now what is agglutination? Agglutination means if particles clump together, then this is called as agglutination. Now in this case what happens, and when antibodies interact with the antigens, then these antigens would stick together in clumps and get particulated. This is called as agglutination. Because of the particulations, because of the agglutination, all these antigens would become immobilized, and once they become immobilized, they would be easily digested by the phagocytic cells. Now such antibodies are called as agglutinins, and the antibodies which performs agglutination are called as agglutinins. See in this figure you can clearly see agglutination of antigens. Now in this one, this is the first dish and this is the second dish. In the second dish you can clearly see presence of blood, but no agglutination has taken place. But in the first figure you can see the RBCs have clumped together. The RBCs have clumped together. Now as we all know, RBCs have got antigens on their membrane. So if we allow them to interact with their specific antibodies, these antigens would produce agglutination, and hence RBCs would particulate, and these produce agglutination. So this is called as agglutination, and such antibodies are called as agglutinins. Now the next mechanism is called as opsonization or coating. Now what does that mean? See, consider this as a microbe or a foreign molecule. Now some antibodies, what they do, they just coat these molecules or these pathogens. They will coat them, they will attach themselves to its surface. Now once they get attached to these surfaces, now phagocytes can easily recognize them. If they are not coated by the antibodies, your phagocytes would take time to recognize this, but once they are coated, they are opsonized with the antibodies, your phagocytes can easily recognize them and act over them, and such antibodies are called as opsonins. So antibodies are of different categories depending on their function. Opsonins produce opsonization, agglutinins cause agglutination, and antitoxins neutralize toxins. See this figure. In this figure, this is a bacterium and your antibodies has opsonized this bacterium. So your phagocytic cells, now these phagocytic cells has got some complement receptors, and these receptors now would easily attach themselves with these antibodies, and then they would easily digest this bacteria. So this is called as opsonization. Now the next topic is types of immunoglobulins. As we already discussed, there are 5 types of immunoglobulins. So before discussing the types of immunoglobulins, I would first tell you the types of heavy chains that are present in immunoglobulins. There are total 5 different kinds of heavy chains that may be present in immunoglobulins. The first one is gamma chain. The second one is alpha chain, the third one is mu chain, the fourth one is epsilon chain, and the fifth one is delta chain, and these are represented by their particular symbols. This is gamma, this is alpha, this is mu, this is epsilon, and this is delta chain. You must have learned about these symbols in physics and mathematics. Now listen to this carefully. Each different type of immunoglobulin has got different type of heavy chain, and the name of immunoglobulins depends on which type of heavy chain they are having. Now the antibodies, the immunoglobulins with a gamma chain, gamma starts with G, so such immunoglobulins are called as IgG. Similarly, immunoglobulins with alpha chain, alpha starts with A, so such antibodies are called as IgA. Mu chain, antibodies with mu chain, mu starts with M, so such antibodies are called as IgM. Antibodies with epsilon, epsilon starts with E, so such antibodies are called as IgE. And delta, antibodies with delta, delta starts with D, so such antibodies are called as IgD. So this is the reason why antibodies are named so. And now if we look at the types of light chains, so there are 2 types of light chains, which are lambda chains and kappa chains, but the naming is because of heavy chains. So let's move ahead and compare 5 different types of immunoglobulins. The mnemonic is GAMED, IgG, IgA, IgM, IgE, and IgD. Let's first look at the structure. IgE, IgG, IgE, and IgD, all of these are having the same Y-shaped molecule. If I talk about IgA, then in this figure 2 molecules of antibodies have come together and they have been joined by a joining chain, so I can say it as a dimer. If I talk about IgM, similarly in this case 5 different antibodies have joined together, so I can call it as a pentamer. So IgM is basically a pentamer of antibodies and IgA is a dimer of antibodies. So IgG is a monomer, IgA is dimer, IgM is pentamer, IgE is monomer, and IgD is again a monomer. Now if I talk about molecular weights, and the unit would be Daltons, IgG is 1,50,000 Daltons, IgA is 3,80,000 Daltons, IgM 9 lakh Daltons, IgE 2 lakh Daltons, and IgD is 1,80,000 Daltons. No need to remember the molecular weights for NEET exams, but the thing that you should remember is which is the heaviest and which is the lightest antibody. The heaviest, as we can rule out, is the IgM with a weight of 9 lakh Dalton, and the lightest is IgG with a weight of 1,50,000 Daltons. Next is their composition. Now what percentage of these antibodies forms the total amount. IgG forms 80% of the total antibodies, IgA 13%, M 16%, E 0.002%, and IgD around 1%. No need to remember the composition, you need to remember the which one is maximum and which one is minimum. IgG is maximum in concentration and Ig is minimum. Next is a very important question, that which type of antibody crosses placenta, and only IgG can cross placenta, none other antibody can cross placenta. So if there is a question that fetus receives which kind of antibodies, so our answer would be IgG. Next is the function. See, IgG is the main antibody in secondary response, as we discussed in our previous lectures about the secondary immune response. In secondary immune response, the main antibody is IgG, and it provides passive immunity to fetus, as it is the only antibody that can cross placenta. IgA is always present in secretions like saliva, colostrum, and mucus. What is colostrum? It is the initial milk that is discharged, that is released after parturition or the baby birth. So the baby gets passive immunity in the form of IgA. Your saliva and mucus protects you by the antibodies IgA. IgM, it is the main antibody in primary response. Remember this, in primary response the main antibody is IgM, but in secondary response the main antibody is IgG. And in any sort of infection in your body, the first antibody that interacts or that produces action is IgM. IgE, very important, it is the antibody that fights against allergy and parasites. Remember, IgE, many times this question has been asked. IgE is the antibody against allergy and parasites. Now here I'll give you a very good mnemonic to learn one more concept. See, there are two E that fights with allergy and parasites. Now first of all, I would make one thing clear. See, basically if I talk, bacteria, virus, fungi, protozoans, nematodes, and even flatworms, all of them are parasites. But here in this case, when I am talking about allergy and parasites, then I am talking about helminths. That is flatworms and roundworms. So IgE would fight against any kind of helminthic infection or roundworm or flatworm infection. So in such parasitic and allergic infections, 2E would fight for you in the body. The first E is IgE and the second E is eosinophil, a type of WBC. Same kind of question is asked for WBCs, that which WBCs fight against allergy. So our answer would be eosinophil. So you need to remember that in cases of allergy and parasitic infections, 2 E's, that is IgE and eosinophil, would help you. And the last one is IgD, which acts as a B cell receptor. So that's it. Thank you very much for watching us. Stay connected with us for such informative lectures. Thank you very much for watching us.